Today, in this lecture, we are going to talk about concepts related to macula densa and its role in tubuloglomerular feedback mechanism and autoregulation of GFR and renal blood flow. So, talking about macula densa are basically specialized cells that are present in the distal tubule of the nephron, and they try to regulate the amount of sodium delivered to the distal tubule. So they are specialized cells specialized in detecting the amount of sodium in their distal tubule, and with the help of detecting the sodium in distal tubule, they are able to do auto-regulation of the GFR and renal blood flow. They are able to maintain normal level of glomerular filtration rate and renal blood flow. And that's the role of macula densa, and which is normally labeled as tubuloglomerular feedback mechanism. So to discuss this thing in detail, how the macula densa basically auto-regulate GFR renal blood flow, we will quickly revise what we have studied in the last few lectures, and then we will come back to macula densa. So quickly revising what we have studied so far. We know that inside the nephron, we have studied so many times, if you have watched my previous lectures, that nephron is the basically structural functional unit of the kidney. It is performing the urine formation process. This is the nephron, this is one nephron, this is another nephron. Blood enters the nephron at this level into the Bowman's capsule, filtration occurs, filtrate moves to the proximal tubule, then the loop of Henle, then the distal tubule. And at the distal tubule level, this distal tubule is in cross proximity with the efferent and afferent arteriole, where filtration is occurring into the Bowman's capsule. And here we have the macula densa. Now, when we cut this area and get a section of this area, we have a picture like this. Here we have the distal tubule, which normally is present here. And here we have the macula densa in the red color cells. And they are in close proximity to the afferent arteriole and efferent arteriole. Now, how they basically regulate the GFR and renal blood flow. In the last two lectures, we talked about the auto-regulation of GFR and renal blood flow, and we discussed that auto-regulation of GFR and renal blood flow simply means that the ability of the kidney to maintain nearly normal level of the GFR renal blood flow despite of changes in the arterial pressure. With the help of this graph and this curve, we explained when the arterial pressure increases, the urine output increases. When the arterial pressure decreases, the urine output decreases. And at the same time, when we increase the arterial pressure or decrease the arterial pressure, there is a big resistance to change in the GFR. This red color curve is showing GFR, blue color curve is showing the blood flow. And it shows that the normal arterial pressure is 100, and at 100 we have this level of the GFR and blood flow. But even if the arterial pressure increases and decreases, still the level of the GFR, they are kept nearly normal. Similarly, the level of blood flow to the kidneys, to the nephrons, it is kept nearly normal. Even with big changes, only when there is a tremendous change, a very big change, only then the GFR will drop and the renal blood flow will drop. But otherwise, the kidney has their own function or their own ability to keep the GFR and renal blood flow despite of changes in the arterial pressure, systemic arterial pressure. You can compare this thing with the urine output. Now, the urine output is definitely affected due to the arterial pressure. The GFR and the blood flow are maintained pretty much normal, almost to the near level. So this is the auto-regulation. Now, to maintain auto-regulation, or how the auto-regulation is done, how the kidneys basically auto-regulate the GFR and renal blood flow despite of changes in the arterial pressure. So the kidney is doing this thing, the kidneys with the help of tubuloglomerular feedback mechanism. And in this mechanism, with the help of changes in the sodium concentration in the distal tubule, the resistance in the afferent and efferent arteriole, they are basically, the resistance is basically either increased or decreased with the changing level of sodium concentration in the distal tubule. So here in the distal tubule, we have the macula densa, and here in the afferent arteriole and efferent arteriole, we have the JG cells, the juxtaglomerular cells. So these two cells are important in maintaining the sodium level, and when sodium level is maintained, the GFR and blood flow are also maintained. So we can say that most of the time the GFR and the blood flow levels are maintained at the expense of sodium level in the distal tubule. And sodium level are detected by the macula densa cells. These are specialized cells, and they give signal to afferent arteriole and efferent arteriole by decreasing the resistance in these arteriole. Now, coming to the point, how macula densa basically affect the resistance in afferent arteriole and efferent arteriole. Now, whenever the sodium level in the distal tubule drops, it may be due to the arterial pressure. Because when arterial pressure drops, the glomerular hydrostatic pressure drops. Now, we have drawn the glomerular capillaries here, and we have shown the glomerular hydrostatic pressure. Here we have the afferent arteriole, here we have the efferent arteriole. When the arterial pressure drops, the pressure in afferent arteriole drops, the glomerular hydrostatic pressure drops, and filtration process decreases. When filtration process decreases, the filtrate moves slowly in these tubules, and these tubules absorb a lot of fluid along with sodium. So the amount of sodium delivered to this point is detected, and it is quite low. When it is detected and the sodium is quite low, the cells, the macula densa cells, they get activated. Now see, the arterial pressure has dropped, the glomerular hydrostatic pressure has dropped, which has decreased the GFR, the glomerular filtration rate. Now this signal, this drop in GFR leads to decrease in sodium level at the macula densa cell. Decrease in the macula densa sodium chloride level. Now this decreased level of sodium chloride at the macula densa in the distal tubule leads to two pathways: the afferent arteriolar feedback pathway, the efferent arteriolar feedback pathway. And these feedback pathways are executed with the juxtaglomerular complex. The juxtaglomerular complex consists of the macula densa cells and juxtaglomerular cells in the afferent arteriole and efferent arteriole. Now, when the sodium level has dropped in the macula densa cells, this macula densa cells send some signals to the afferent arteriole. It sends some signals to the afferent arteriole, and it basically tries to decrease the resistance at the afferent arteriole. The resistance in the afferent arteriole decreases. Now, when the resistance in the afferent arteriole decreases, what happens is that a lot of blood starts coming through the afferent arteriole into the glomerular capillaries. The glomerular hydrostatic pressure increases, filtration increases, and the GFR returns to normal. Now this is one way, by decreasing the afferent arteriolar resistance. Another pathway, now this macula densa has some special secretory substances which go from here, from the macula densa cells to these afferent arteriole, and with the help of those substances the resistance increases. But it also starts another pathway, the efferent arteriolar feedback pathway, in which it releases renin. It sends signals to the efferent arteriolar cells, where the juxtaglomerular cells get activated. These are the juxtaglomerular cells. They get activated and they secrete renin. Now when renin level increases, it converts angiotensin 1 into angiotensin 2. It acts as a hormone which basically converts angiotensin 1 to angiotensin 2. So when angiotensin 2 is increased, the level of angiotensin 2 when increased, and with the help of renin, this angiotensin 2, it basically increases the resistance. It increases the resistance at the efferent arteriole. Now if you see the macula densa, the specialized cells in the distal tubule, they are decreasing resistance in the afferent arteriole, but they are increasing resistance in the efferent arteriole. Now what happens is that due to angiotensin 2, renin was released from the juxtaglomerular cells, and due to renin, a lot of angiotensin 1 is formed, and then angiotensin 2 is formed, and this angiotensin 2 increases resistance at the efferent arteriole. This is the efferent arteriole, and resistance increases, so this pressure builds up, and again due to increased resistance, the glomerular hydrostatic pressure increases and the filtration increases. Now we have seen here two things, that decreasing the resistance at afferent and increasing the resistance at efferent with the help of angiotensin 2. Decrease resistance at efferent and increase resistance at efferent will increase glomerular hydrostatic pressure, which will increase the filtration process, and a lot of filtration will occur. And a lot of fluid will start coming back, a lot of sodium will be coming back, and the sodium level detected at the macula densa at this level will be normal, and this tubuloglomerular feedback will end, and with the help of increased efferent arterial resistance and decreased afferent arteriolar resistance, the GFR will return to normal. So that's how macula densa is playing an important role in tubuloglomerular feedback pathway, which is trying to return the GFR and the renal blood flow to normal level, which is basically the autoregulation process. Now, if you remember, or if you have watched our previous lectures, we discussed in detail that these curves show the efferent arteriolar resistance and the afferent arteriolar resistance. It is showing the GFR and it is showing the GFR and blood flow. So at both levels, when the afferent arteriolar resistance increases, the GFR decreases and the blood flow decreases. But when resistance at the efferent arteriolar when the resistance at efferent arterioles increases, initially there is an increase in GFR. So decreasing resistance at the afferent arteriole, will increase the GFR, but decreasing resistance will increase the GFR. Increasing resistance at the efferent arteriole increasing resistance at efferent arteriole will initially increase the GFR, but if the resistance increases too much, then the GFR will drop. So that's how it has an effect. That's how it has an effect on the glomerular hydrostatic pressure. That if the resistance at this site increases and resistance at this site decreases, the glomerular hydrostatic pressure will increase, the filtration process will increase, and the filtrate in the tubules will increase. So when the GFR increases, then it is restored, and that's the role of the macula densa in this process. And we were here to discuss the concepts related to the macula densa, that how it is basically playing its role in tubuloglomerular feedback or the autoregulation of GFR in renal blood flow, how these simple cells present in the distal tubule can restore the blood flow to the kidneys and GFR. Now, the mechanism, the tubuloglomerular feedback mechanism may not be 100% perfect, but it plays an important role. If there is a slight decrease in the arterial pressure, and slight decrease in the blood flow, then this can be recovered with the help of the tubuloglomerular feedback pathway. But if a very big decrease in the arterial pressure occurs, for example, there is a very big huge amount of blood loss due to a very a big accident, then the blood loss may be so much that these pathways won't be able to play their role, because there won't be any sufficient time. The bleeding will be so much, or the dehydration will be so much, or the fluid loss will be so much that these cells won't have any time. Rather, they can further deteriorate the renal shutdown in such conditions, because in those conditions the GFR can be brought back very rapidly down and the renal shutdown can occur. So they have their role, the macula densa and the tubuloglomerular feedback have their role in the autoregulation of GFR and the blood flow, but there are certain limits. In those limits, it is performing their functions. So that's all about this macula densa cells, and in the next lecture we will see that how different drugs used for the renin-angiotensin pathway can basically be used for treating the high blood pressure and bringing down the arterial pressure. Thanks a lot for watching the video.